The second type of cells which originate from the myeloid lineage are the agranulocytes. They are also called antigen presenting cells because they take up the antigen or microbe, digest it, and present it on its surface. These cells are generally considered as a bridge between innate and adaptive immune response because at one end they make contact with the antigen and on the other hand they interact with the lymphocytes which are present in the lymph node to activate the adaptive immune response. These agranulocytes also secrete protein which attracts and activates other immune cells. So basically there are three different types of agranulocytes: monocytes, macrophages, and dendritic cells. Let's discuss them one by one. So monocytes are generally mononuclear, having one nucleus, which is generally bean-shaped. These monocytes generally circulate in the blood for about 8 hours, during which they enlarge and they migrate to different tissues and differentiate into specific cells. So these monocytes are considered as the precursor cells of tissue macrophages or dendritic cells. So macrophages are also mononuclear cells which originate from monocytes when they reside in specific tissue uh, and mature into macrophages. Macrophages are the primary phagocytic cells in the immune system. They easily take up the bacteria or antigen by endocytosis and digest it with the help of lysosome. These lysosomes contain different hydrolytic enzymes which fuse with the phagosome to form a phagolysosome, which digests the content of this lysosome and eliminates it by exocytosis. So these macrophages also have the ability to present some part of the microbe on its surface as antigen. So that's why they are called antigen presenting cells. The phagocytic activity of the macrophages can be enhanced by a process called opsonization, in which the antigen is coated by either by antibody or certain complement component, which makes macrophages more attractive towards them and enhance the phagocytosis. The macrophages can be present in a free form or reside in a specific tissue. When they are residing in a specific tissue, they are called fixed macrophages, and the name changes depending upon the type of tissue they reside in. For example, when they are present in lungs, they are called alveolar macrophages, or when they are present in the liver, they are called Kupffer cells. The third type of agranulocytes are dendritic cells, which have these branching shaped cells or star shaped, and they are also the antigen presenting cells because they capture and present antigen to specifically to T lymphocytes. These cells can be present on non-lymphoid areas as well as lymphoid tissues like lymph nodes. The lymphoid progenitor cells then differentiate into larger lymphocytes and smaller lymphocytes. The larger lymphocytes then develop and mature into natural killer cells. These are the cells which have granules in the cytoplasm and are part of the peripheral blood lymphocytes of human. So these cells are involved in providing innate response to viruses and other intracellular pathogens. So they can also recognize tumor cell either by a non-specific or direct method or by a health antibody specific method called ADCC or antibody dependent cell mediated cytotoxicity. We will discuss this phenomenon in detail in our upcoming lectures. The other type of lymphocytes, that is the small lymphocytes, then differentiate and develop into B cells or T cells, which are part of adaptive immune response. Generally, whenever we say lymphocytes, we are generally talking about the small lymphocytes or B or T cell and not the natural killer cells. B cells are named as B lymphocytes based upon the site of maturation, that is bone marrow in humans and bursa of fabricus in birds. So these B cells can be differentiated from other lymphocytes by the presence of a membrane bonded immunoglobulin or antibody. B cell when activated um, develop into plasma cells which secrete antibodies, and at the same time they are also able to produce memory cells which will remember that antigen or microbe for future response or secondary response. The T cells are called T lymphocytes because of their site of maturation, which is thymus, and these cells also have receptors for antigens like B cells, but they do not have any antibodies attached to the membrane. Now let's look at how the different types of lymphocytes work. The B cell lymphocytes or B lymphocytes are the one which have B cell receptors on their surface. These receptors are antibodies. So these cells can bind to any antigen or microbe. Not essentially, it has to be binded to some specific cells. So, by the activation, antibodies, plasma cells are produced, which release antibodies, and these antibodies function by neutralizing the microbes, or by they can generally do phagocytosis or complement activation. We'll discuss complement activation in our future lectures. So T lymphocytes or T cells are of three types: helper T cells, cytotoxic T cells, and regulatory T cells. So T cells are generally different from B cells by the fact that the receptors on the surface called the TCR are not antibodies, and they can only bind to those antigens which are presented by antigen presenting cells like we have discussed earlier. So the helper T cells, when they are activated, release some chemicals called cytokines, which results in activation of macrophages, inflammatory response, or activation of other T and B cells. The cytotoxic T cells or T lymphocytes also bind to microbial antigen presented by the APC, antigen presenting cell, and results in directly killing of the infected cell in which this microbe is ingested. The third type of T lymphocytes are called regulatory T lymphocytes that results in suppression of the immune response. The third type of lymphocytes are natural killer cells which directly kill the infected cells. So it's time for our second checkpoint. So you are given different questions and you have to give answers to each of them. For example, in the first one, you have to find out the adaptive immune cell that matures in the marrow. The answer can be single type of cell, or multiple types of cell depending upon the type of question asked. Try to answer these questions and we'll discuss the answer in our interactive session.